I'm calling to order the 1242nd regular meeting of the Entomological Society of Washington. Welcome. And the first order of business after you pay for your pizza, if you're present here, whether using the Venmo or the
This meeting is being recorded.
please, is the reading and approval of minutes by Secretary Gary.
Thank you, Don. The 1241st regular meeting of the Entomological Society of Washington was called to order on the 4th of January 2024 by President Don Weber at 7 p.m. in room WD 33 of the National Museum of Natural History. 21 members and 5 guests were in attendance with 22 virtual visitors. Recording Secretary Gary Hevel read the minutes of the December meeting, which were approved after a few suggested changes from members of the audience. Lourdes Chamorro noted that members who renewed their memberships electronically last year will be automatically renewed for the present year. Floyd Shockley introduced Dr. Jeremy Frank, a new employee of the Department of Entomology. Program Chair Alan Norbaum mentioned that he found ESW t-shirts from 2015 and is turning them into the society. He also passed around a recent pest shrift issue from a European journal, Studio Entomologia, honoring Chris Thompson. Hannah Litwack provided the cover art for the issue. President Weber announced the death of Mary Cornelius, a colleague who worked mostly on termites at the Agricultural Research Service in Beltsville. She passed on the 7th of December, and her obituary can be found in the latest issue of American Entomologist. President Weber also noted that Bridget Bradshaw from Warrenton, Virginia, recently appeared via Zoom, noting her current study of the diet of kestrels. She is seeking help for identification of insects found in the gut contents of these birds. Someone suggested utilizing iNaturalist for assistance. Floyd Shockley mentioned that the MMNH exhibit and a northern giant hornet has been removed from exhibit, but a mini exhibit of the subject can be observed for the time being at the Hymenoptera section area, 5th floor, East 4th. Program Chair Alan Norbaum introduced the speaker of the evening, past President Matt Buffington, whose topic was From Ordinal Level Phylogenomics to Cryptic Species, 10 years of research and adventures in Hymenoptera. Dr. Buffington offered a review of his career with the Systematic Entomology Lab, dividing the discussion into 6 parts. The phylogenesis of all Hymenoptera was presented, emphasizing the importance of gaining stable phylogeny. Since 1997, more data and better taxon samples have been pursued. Parasitoid wasps emerged in time much earlier than thought, with origins now going back to the Triassic period. Research with a well-curated collection has strong advantages. Important taxonomic studies have now doubled the number of families in the Hymenoptera. Still, there is confusion in the construction of world families. Dr. Buffington has most recently analyzed the cynipids of the genus Anaspis, which includes 67 described species. Standard analyses suggested that there are 60 to 85 species involved in the genus. 2 new species are being described from SODA analysis. A helpful new character has been found on ovipositors that aid taxonomy. Efforts are being made to digitize type specimens, and this work is critical toward a proper understanding of Hymenoptera taxonomy. The meeting was adjourned at 8:45 p.m. Your turn. Yes.
Gary, the Mary's obituary will appear in a future issue, not already.
All right. Yes.
2 things. Ganaspis is I fidgeted, not a cynipid wasp.
Okay,
It's the sister group.
yeah,
And the digitization of types is critical for all taxonomy.
Yeah.
Period.
sure.
Thank you.
Exclamation point.
Exclamation.
The edition honoring Chris Thompson was in Studia Dipterologica, not Entomologica.
Okay, I couldn't remember.
Problem.
Someone online? No? Is that your hand raised? Oh, no.
Just a head scratch.
Has everybody signed it? Is there anything in the chat?
So I think that nothing on my.
Okay. Thank you, Gary.
Yeah, sure.
I wanted to report to the group that we had an executive committee meeting last Monday, January 22nd.
Don?
Yeah,
I think we have to approve.
Yeah.
we have to approve the minutes. I'm sorry. I thought Gary asked for the approval of the minutes.
You have to make a motion.
Are you making a motion to approve the minutes?
Yeah.
Seconded.
Gary, excuse me. Is there any discussion? All those in favor say aye.
Aye.
Any noes? Any abstentions? The minutes are accepted with corrections. Sorry about that. I just want to report from the executive committee meeting a few items, and one of which concerns us, at least one of which concerns us tonight. So our newly elected co-editor of the proceedings of the Entomological Society of Washington, Jerry Cook, is working on the future issues, which include the issues of 2023. And he's working with Mark Metz on this. And there are at least 2 special issues in the works. Yeah. 1. Oh, and possibly a special publication. Yes, thank you. All right. Did you want to say something about the merchandise page that we're
I could,
proposing?
yes. This is Lourdes tomorrow. Through the website, there will be a merchandise page where you can get shirts, hats, maybe bags, and if anybody else has ideas for something that they'd like to see.
Some sweet mugs.
And yeah, mugs, there'll be mugs.
Yeah.
And which one?
Yeah, stickers.
Oh, I don't have stickers.
Stickers.
Okay.
People
And
love
mostly it's
stickers.
just gonna be the logo right now, but if there are other ideas, then we welcome them. And we're going to earn 10% of the cost of whatever we put there, but we want to test the quality, so we're going to make a small order just to make sure that it's doesn't disintegrate in the first wash. So those will be available in person, but then you just pay for them through the website, and then they'll be shipped directly, so we don't have to store them.
All righty. Do we want to say anything about the new tranche of found t-shirts or no?
I think we can buy them here for $12.
Okay.
Yes.
Apologies to the online participants. Yeah.
Going back to memoirs and special pubs, I was
Yeah.
able to form a special publications committee chaired by me. And include Norm Woodley and Tom Henry. And the 3 of us are going to try to hammer out exactly what a memoir constitutes, what a special publication constitutes, and hopefully get some parameter space up on the website so people kind of have an idea.
Yeah, so we can solicit and they know
That's
what they're
exactly right.
applying
So
for.
we're gonna go full steam ahead.
Excellent, thanks. Food for meetings, that was the one I thought concerned at least those present now. I guess I'll kind of throw open the floor to any discussion. You know that currently we are ordering Pizza, Matt, thank you very much. That is with society funds, but we hope that to fully defray that by combination of the Venmo QR codes and the donations and the up with cash. And you're always welcome to bring stuff in. I brought in some guacamole and somebody brought in some prohibited liquids and see, well, whatever. Anyway, are there any comments on the food? We have 3 more spring meetings that you do or don't like, Dave?
Just a little history. When I
Yeah.
was here in the early 90s, which Gary remembers this too, each meeting they would ask for volunteers to bring the refreshments the next meeting, which I thought was a little strange given experience at other societies. So we then dumped it on the program chair. For many years, the program chair or chairs, because John Brown and I shared that for a while, would bring the refreshments. So I guess now there's yet a 3rd iteration of refreshments, but it's just a little history.
Okay, are you satisfied with what we do now?
I haven't been to that many meetings.
Okay,
Partly because I'm out of
any
it.
other input?
I like the way things are now.
You like?
Quick question.
Yeah,
So, next month is my birthday and I was thinking of bringing prohibited liquids, but, what, are they gonna check my bag on the way in if I bring them in?
I very much thought, I
No.
brought a whole thing of guacamole and chips and.
Okay.
Yeah, no, it isn't looking
It's
at-
easy to bring stuff in. It's harder to get stuff out.
Okay.
take a very
Just don't run so the bottles don't clank.
Yeah.
So see, at some point though, I remember like when I first joined, we were all going out, like we were meeting at like
That's right.
5:30
Yeah, that's
and going to a
true.
local restaurant.
For a while, too.
That was very stressful.
It
Yeah.
was.
Okay. So we're.
I'm not
okay.
suggesting that. I was just to
Okay.
provide additional
That's true.
context.
All right, we're gonna, yeah. Matt.
To keep it as it is now, I think we need to do a financial readjustment of what each slice costs.
Yes.
Okay,
So I will make that calculation based
Yeah.
on tonight's, and
saying,
then we'll send that out, and that's gonna be kind of like how it is.
okay.
Yeah.
And so you honor system, you put in what you make. All
And I
right,
think financially things are going to go better with the demo.
Yes. Absolutely.
okay. Great,
Oh, and Treasurer Kula asked that if you can make a note when you make the payment, pizza. It'll help her. with-
like as opposed to
Yeah,
something else.
something else, yeah.
All right, great. I think that does the discussion. Apologies to our online participants that you're not able to enjoy the food tonight. Maybe you're enjoying something else. One more item, and that is, I believe Talita is organizing the room with the proceedings issues, the hard copies on Friday the 9th. Is that true?
We're gonna reschedule.
Yeah,
What?
I'm gonna be I was gonna help her and I'm gonna be out of town.
Okay, so you're gonna reschedule it.
Yes,
Yeah.
Okay, but if you can let people know, so you get some help, right?
yes.
Okay. I just had a note that you're doing that.
You know, there's more of those up in the attic too.
Oh,
Like
One.
almost
space
exactly
at a
the same
time,
amount
but-
is up in the attic.
To protect that, that room has bulletproof, that glass, and that room is bulletproof.
And Dawn, just to say, so we're going to digitize the first 3 issues of the members
Okay,
and make them available to membership
Mhm.
through the website,
okay,
with Talita's help.
Yep.
So that was something we discussed and that we were going to provide. So the first, that are out of print, the first 3 memoirs.
I had that down. That was my next item.
Oh, so sorry.
I also have to note that I need to form an audit committee to review the books as presented already by Treasurer Abby Kula. That will be done shortly. Then I appreciate volunteers. Oh, you're doing it.
Oh, no, I have a comment about the pizza.
Yeah.
Venmo, which is that this is not an actual Venmo QR code. It go to your phone app, not to the Venmo app, because it does not work in the Venmo app. It's a different app.
Oh, okay,
It works, just not through, it's not actual Venmo.
okay. So noted, I guess.
Just go to your phone. Yeah.
I'm gonna call on President-elect Andrew Jensen for any banquet news, et cetera. Any updates on that?
Sure, we're thinking about the Log Lodge in Beltsville as a location for this summer's banquet. Still working out the details for reservation and speaker. That's all the updates for now.
That is not close to the metro, but we could easily set up a shuttle from Greenbelt. And it's been rather thoroughly revamped. If you've seen it before that, you might, but it's a renovated building. So thank you, Andrew. Do any other officers have any reports? Hearing none, I'll ask for introduction of new members and visitors. First here. No? Yes?
I'd like to introduce a visitor. In the blue sweater over there is Austin Fife. He's a new postdoctoral researcher in the electron and confocal microscopy unit in Beltsville. ORISE.
Excellent.
Welcome, Austin.
Welcome, Austin.
Glad to be here.
Any other new members or guests? And online, I don't know quite how to, do we have any folks online who want to introduce themselves? I see Steve Panosian.
It's Stephen Panosian.
Sorry.
I've been a virtual member for since before the pandemic. In fact, I would be there with you tonight, but I have a very nasty cold and I don't want to get anybody sick. So enjoy the pizza. I've been involved with the state of Maryland's mosquito control since 2013. And so I have a big interest in arbo vectors in general. And I'm excited about hearing insects underground tonight because apparently mosquitoes now are adapting and changing their behavior and going into sewers and things. So
Parking garage.
Factors into it.
Thank you. Anyone else online? After we do any unfinished business or new business? Pause, pause, pause. Could I have presentation of notes and exhibition of books or specimens?
Okay, yes, I have one. I have a book called Beetles of the World, A Natural History by Max Barclay and Patrice Bouchard. If you want to take it over there, thank you.
This is Princeton University Press, and it appears to be full color. And since somebody on high had an inordinate fondness for beetles, I recommend it. I do too.
That same series also has, I think, Bees of the World, Hymenoptera of the World. There's one, Velvet Ants of the World is coming out soon.
Wow.
So nice. Okay, I have one here. It doesn't sound entomological, but it is pretty much that. It is Tree by Tree by Scott Miners. It's new. This is from the Arlington Public Library. Saving North America's Eastern Forests. And what it does is it looks at elm, chestnut, hemlock, ash, sugar maple, and some other trees, and ones that have, what shall we say, challenges, at least half of which involve insects, like emerald ash borer, like hemlock woolly adelgid. And some of our entomological colleagues, particularly those involved with biological control, are cited. It's a well-cited nonfiction book for the general public, and I think a really good one. I'm halfway through it. Any other exhibits? Yeah. Hey.
Yeah. Well, in honor of our speaker, I brought this. This is a paper wasp nest from
This
Brazil.
way, go there. See.
Oh,
So they can see it.
where?
There you go.
There, you're in the, yeah, there you are.
Okay, this is a paper wasp nest from Brazil, adorned with some local seeds and a piranha, part of a piranha jaw from the Amazon. Apparently, it's a very common tourist thing in certain parts of Brazil. And I have it on good authority, and this is the relation to tonight's talk. Jim Carpenter, who was the advisor of our speaker, and also the undergraduate advisor of that crazy guy with the long gray hair over there and beard, Paul. But anyway, Jim told me it was probably, it's Charturgus is the genus, probably Artiflex. And they're pretty aggressive paper wasp, but the nest is pretty hard. So I just thought I'd bring this to show you. And it relates to both Hymenoptera and our speaker.
If people buy them just like as a cool bauble, like.
Yeah, yeah.
I know, right? Yeah.
And it's adorned by the local, the indigenous people with the various seeds that are local, I recommend it.
Wait, is this something that you own?
No, it like a lot of things that come to the department, like Floyd in his office has a whole drawer of the beetles from Merida, Mexico, which they adorn with rhinestones and a chain, so it's live jewelry.
So this was intercepted.
I don't know that this was intercepted. I'm not sure. I don't know the history of it. It's just that the old Ashmead room, which you probably have heard of but never saw, this was hanging there, and they were going to throw it away. So now it's hanging out in the sorting center.
Another artifact found.
Yeah.
Thank you, Dave. Any other exhibits, show and tell? Yes.
A comment on that. In Microlepidoptera, we have a Charturgus nest that Microlepidoptera was reared out of.
Well, there's paper inside, so who knows what might be breeding there.
Yeah.
I mean, the paper nest,
Uh-huh.
probably domestic.
I'm gonna call on past, unless we have any more exhibits, I'm gonna call on past President Matt Buffington to introduce our speaker tonight.
Thank you, current President Weber. It's my pleasure to introduce Dr. Jeremy Frank, who's going to be giving our lecture tonight. Thank you so much for kind of filling in last minute, but you weren't a last minute thought. I was thinking, I suggested you to Alan a long time ago, so. Jeremy got his PhD at the Gilder Graduate School at the American Museum with Jim Carpenter, and then took a position as collection manager at the Bishop Museum. And then one day I saw your name floating around in some correspondence here. I'm like, I think he's now at Smithsonian, so that's super cool. And I know Jeremy, he was a Hindcourt student in Hindcourt's 2016 North Carolina, right? Something like that. So I'm just super excited to hear your talk tonight. So without any further ado, take it over, Jeremy. Cece will help with the tech here.
Sounds good. Let's see what happens when we get to the videos. Those never work. So, hi everyone. I'm Jeremy Frank. Today I'm presenting the Curious Troglodytes, Insects Underground. Kind of a two-part talk, united by the excitement of insects that are below the surface. Here I am. So, we've already heard a little bit about me, but I love this picture, so I take every chance I can get to show it off. I like to think I look very inquisitive. No big bosses there, though. It's a shame. So I've been at Smithsonian for about a month and a half. Prior to that, I was in lovely Hawaii at the Bishop Museum as collections manager. But the one thing you can't get in Hawaii, you can get fantastic weather. There's a beautiful collection out there, but they don't have native sand wasps, which are really my passion. You can see me here in this photo. That's after finding my first sand wasp in the wild. Very, very happy. So the first part of my talk, I'll be talking a little bit about those. And it's not really a theory that I'll be discussing in this talk, but I do sort of like to think that entomologists resemble their study organism a little bit. You can see if you agree with that. Look at those big toothed. So the first part of this, I'm calling solitary wasps, the other Hymenoptera. So not these, which would be some of the famous social Hymenoptera, of course, living in fantastic nests and cooperatively raising the brood. Everyone knows a little bit about those. And not these, which are parasitoids and doing very economically important things. Not that I'm a little bit jealous about that. Certainly not these, which get enough attention, other social Hymenoptera. But something like this, you may have seen one of these. Somebody may have sent you a picture or a specimen and said, oh my God, it's a murder hornet. It is not. This is a lovely cicada killer, a solitary wasp. I was going to say quite gentle, but not towards cicadas. But there's actually quite a large number of solitary wasps, Aculeate Hymenoptera, for 20,000 species, Apoidea, Vespoidea. They have a tremendous diversity of form and lifestyle. And can't talk about all of them, but I'll try to give a little bit of a broad overview. So unlike the social wasps, which tend to construct nests, for example, out of the paper that we saw earlier, that was a really great example that I wasn't planning on, but I do appreciate it. They tend to excavate nests out of the soil or create small nests out of mud. Sometimes they occupy existing structures, hollowed out twigs, disused nests of other structures. I was wandering around the sculpture garden nearby on my lunch break. I saw a mud dauber nest. I got very excited. So the reason they're called solitary is that they're always raising their brood independently. There's no cooperative care, none of that lovely advanced eusociality that you hear so much about. But they do sometimes occur in high density. We'll see if this works or causes everything. Well, great. So you can see this would be a sand wasp nesting site. You can see them flying around. So while they're solitary, they're by no means occurring necessarily very far apart. So all of these wasps have their individual nests, which would be concealed underneath the surface. So video of the slide. How do I get to that next slide? Here, I thought I was doing so well with that. There we go. So you can see here an even further example of interaction between solitary wasps. So we have some lovely benthics, and they're fighting over a paralyzed fly. So while they may be solitary, they are still interacting. They're just not cooperating. So, lovely little bit of nature bred in. So talk a little bit more about their provisioning. So they tend to stock their nests with paralyzed prey. So the prey, at least in the group of wasps I'll be talking about, is still living. So they'll be stung, often attacked on the wing, and then they'll be stocked away in these nests for a singular larva at a time to consume its own individual provisions. So this provisioning comes in multiple types. It may be all at once, where the female wasp will stuff a nest full of all of the prey that her larva needs for its entire development and then seals the nest off, or progressive provisioning, in which food is brought in a little bit at a time over the course of the larval development until it's almost ready to mature. And the females in some species can actually assess the larval development, so they can determine based on the size of the developing larva how much food they need to bring in that day. So even though they're not living in this complex social structure, they're doing some pretty complex things. And so one of the really interesting things in sand wasps and bembics, which are the group that I'm talking about, is that they have a little bit of host specificity, but not entirely. So in bembics, they'll attack Diptera almost always. And they'll attack any Diptera of the appropriate size. It doesn't really matter whether it's a sarcophagid or a mucid. They'll go after it as long as it's a fly of a size relatively well suited to the developing larva. But they won't attack other insects that are roughly the same body shape and size. Or actually, they'll attack them, but they won't finish stinging them. They'll sort of bump into them and say, hey, that's not right, and move on. So they do have this host specificity, even though they're actually attacking prey. The adult wasps don't actually feed directly on the prey themselves. They tend to feed on nectar. But what this wasp is doing there, it's actually feeding on the gut contents of that fly. It will use the fly's body to feed its larvae, but it's actually getting a little bit of an extra bonus for the mother feeding off the gut contents. So as a result of this sort of life history where they're attacking things like a predatory wasp, but they have this host specificity like a parasitoid wasp, they're sometimes called predatoid wasps. If you were here last month, you might have heard that term in Matt's talk, which really excited me because you don't actually hear that term too often. It's a really great term. It's accurate, but it sounds a little clunky. I remember being told by my advisor that I probably shouldn't use it too much because it never caught on. I'd like to change that. Please use predatoid as much as possible. But the common term is indeed solitary, which, like I've already said, is a little bit of a misnomer because they can occur in really large densities. They are interacting with each other, not just for mating, but for competition. They're just not cooperative. So really a better term might be selfish wasps. And I think the thing is that these solitary wasps don't always get as much attention as social wasps or parasitoid wasps, or especially bees. It's a little bit lonely, but I think they really deserve some attention because they do some really cool things. And the group that I've chosen to dedicate myself to are bembex, also known as sand wasps. There are other wasps that are also known as sand wasps. They're part of a group called digger wasps. The common names get pretty messy, but it's a fantastic group with a worldwide distribution, which is unusual because all of their close relatives are restricted to North and South America. But bembex is spread across the world, and it's actually achieved its greatest richness in Australia and Africa. And they are specialists, as the name might imply, in sandy soils. And so we've already had 2 videos work. I'm hoping this third one will. So you can see here, this sand wasp going to work. This is a female of the common North American species, Bembex americana. She's uncovering a nest that she's already dug here. So there's a soil plug at the top of that nest, and right here she's excavating it, being dive bombed by another wasp, so that's solitary. But they dig out these nest structures, these tubular nest structures going down for 30 to 40 centimeters, terminating in an ovular cell. Each cell will contain 1 egg and the food needed to provision it. They're Diptera specialists. The progressive provisioners, meaning they're assessing the state of the larva. They're bringing in a little bit of food every day until the larva is almost ready to mature. They tend to be active during sunny periods. And for the 330 species of bembex, that's the general life. Except as it turns out, bembex species actually violate all of these supposed ironclad rules for bembex in various different ways. So they have all sorts of weird additional nest structures that get added to this seemingly simple burrow. There are things called back burrows. There are back furrows, which are different from back burrows. There's outer closures. There's inner closures in the nest. There's spurs going off of the nest. They may have multiple cells so that once one larva finishes its development, another cell can be dug out of that nest without the female wasp having to dig an entirely new nest. There's differences in the timing of construction. There's also differences in the provisioning. So almost all of them are Diptera specialists, but there are some that have gone on to Neuroptera, damselflies. In Australia, some of them attack other Hymenoptera. There's a weird, possibly incorrect record of them attacking Lepidoptera. But if you can think about something that usually attacks flies attacking damselflies, that's quite a difference in body structure. So there's a lot of weird things going on. The provisioning isn't always progressive. Sometimes it's mass. Sometimes it's a sort of semi-mass provisioning where the female wasp will start doing it a little bit gradually, and then at the end she suddenly gets a bug up her butt and fills the nest all at once and seals it and goes away. Some of them are active at dawn and dusk instead of during the day. So every species that we've studied of these 330 has their nesting biology in some way unique. So as an early graduate student, one more slide. Morphologically speaking, they tend to be fairly compact. So we'll talk a little bit more about this later on, but most variation is in males. The females are distinguished almost entirely by color and pattern. The color and pattern also tends to pretty much always look like this. They have a sort of a thick U-shaped marking. their various waves of yellow and black. So even when you're trying to distinguish them by color, they all sort of look like that. So I thought, aha, an opportunity. So there's this group with all of these species. They do all sorts of really interesting behavioral things, but there was no phylogeny for them. There were excellent behaviors or excellent publications on the behaviors and the taxonomy, great collections. So as an excited first-year graduate student, I thought this will be my group. So my goals were to build the first phylogeny for Bembex, figure out why their behaviors are so diverse and how those behaviors evolved. So the first thing I decided to do was to try to optimistically build a phylogeny based on that behavior. I thought, well, the morphology isn't supposed to be all that distinctive, but the behavior really is. So let's see what happens if we use the behavior as a data source for a phylogeny. So most of these behaviors are related to the nesting and provisioning. That's where most of the diversity is. So for 34 species, mostly North and South American, but with some European and Australian, those were the ones that I assessed had been described well enough in the literature to score these characters, which is about 10% of the generic diversity. And I got this first year of grad school. So it's not fully resolved, which was a little bit disappointing, but I decided to do what I could with it anyway. So there were several issues with the behavior. I was a little bit optimistic. One reason that you weren't resolving this all the way is that the taxon selection was somewhat uneven. It was driven primarily by what there was data for rather than taking a whole genus approach. It also turns out there's a ton of behavioral diversity within species, things where there would be nest structures that some populations would use and not others, or things that would sometimes be added, sometimes not. It's also possible that behavior doesn't vary significantly within species and that there's unrecognized diversity within Bembex, as I discovered after scoring one very nice Bembex species and then finding out it was actually 2 species, and behavioral differences actually correlated pretty well with that, which was exciting, but I had to redo some of that work. So I said, okay, I'll go on to morphology. And looking at the morphology closer, some of the things that have been published on that were a little bit interesting. The first thing that I'd read was that the genus was compact and not really divisible into clearly defined species groups on the basis of that morphology. But also in the lovingly called Big Blue Book of Specid wasps of Bembex, it was said that there was such diversity in structure that it was difficult to characterize the genus. So obviously a genus can't really be morphologically lacking and stunningly morphologically diverse at the same time. So that was sort of a question I had to ask myself, comparing Bembex wasps that look like this. And so it turns out that, well, the general body plan is the same. A lot of the color and patterning is the same. Especially in the males, there's a lot of morphological diversity going on in some of the finer structures. These are the prongs coming off of the abdomen, which you see in some males, and they have this really beautiful diversity of structure. In the bottom right, there's this beautiful hook coming off of this species. There's little spikes. There's these 2 different forks coming off. Some don't have this at all. So this is a really good structure for distinguishing species. It's also, as far as I know, nobody knows why they have these prongs coming off of the abdomen. So that's a fun question I got to ask myself. So ultimately, it turns out that sort of both of these things are true. In the broader sense, the morphology and coloration and body plan are extremely similar. But on the smaller scale diversity, especially in the males, there's all sorts of things going on. So it did look like there were sort of this reverse mirror of behavior and morphology, where males were morphologically diverse, but behaviorally very compact, because most of that behavioral diversity is going on with nesting, whereas females were behaviorally diverse, but morphologically very compact. And it turns out that taxonomically speaking, a lot of this morphology is useful for distinguishing these sort of groupings of species, but not for determining relationships within those groups or between those groups. If you're interested in Bembex species groups, someone finally went through and published all of those groups together in one place rather than in 100 different years of people sort of coming up with them and forgetting about them. That's one of my papers. You can look it up. I'd be glad if you did. So I took this and I went for a morphological phylogeny. And I was able to include many more species because it's a lot easier to look at a waspinae collection and figure out what its morphology is like than to try to figure out behavior. I would have loved if I had the time and funding to study behavior for 114 species. I did not. So these 114 species, it's about a third of the group's diversity, but it represents almost all of the proposed species groups, 51 out of 54, as well as some that were never formally grouped into species. There were 63 morphological characters in this, about 37 of which are found only in males. So it really is about half a male phylogeny. So you can see that there's some of these characters start to get really, really diverse. There's 8 different states for this prawn coming off of the propodium. There's another prawn elsewhere further down, all sorts of things going on. So very excited. I did a phylogeny using that material, and I got something that did look very nicely resolved, very exciting. But under resampling, kind of tended to collapse a little bit. So that was a little bit of wind out of my eager young graduate student sails. So there were some things to take a look at. So some of these species groups that had been proposed in the taxonomic literature were being recovered with moderate support in this phylogeny. So that was giving me a good sense that there was some signal going on in the morphology. But there was weak support below that group level and above it, which pretty much agrees with what the taxonomic literature had been saying, which was that we can use Bembex morphology to figure out these groups, but not so much at any other level. And also, if we take a look at the behavioral phylogeny and the morphological phylogeny, they don't necessarily agree for the species that were able to be included in both. So while morphology has greater resolving power than behavior, the signal was very different, and neither source was entirely satisfactory. Part of that for behavior is that it was very interspecifically diverse, again, maybe, like I said. And morphology sort of is in this unusual state where it's got enough diversity for certain scales, but not enough for others. So I said, all right, I'll bite the bullet and go to the molecular phylogeny, which I didn't really want to do because I like playing with the wasps much more than I do crushing their legs and playing with colorless liquids for a while. So I went to museum specimens to try to extract this DNA. So I was going for an ultra-conserved elements approach, extracting from historical specimens with an average age of 40 years or so. And had successful sequencing for 28 species, all told. And so once I actually did the genetics, this actually does turn into a nicely resolved phylogeny with much better support. So that was very exciting. And so I won't talk about the entire phylogeny, but I'll talk a little bit about some of the exciting results that were coming out of that. And one of them is that the Belfradiae group was recovered, the species that could be included in the molecular phylogeny, with good support, and it came out where we expected it to come out. It's been proposed as the most basal Bembex, and that's because of their morphology and their behavior. So most Bembex, one of their distinctive characteristics is that the ocelli are obliterated, reduced to stars. But in the Belfradiae group, many of those species have the ocelli partially preserved, and that's probably related to their behavior, which is where these species tend to be crepuscular. So they're active during dawn and dusk as opposed to during bright daylight hours like all of their relatives. So they need to preserve those ocelli to see better in low light conditions. So this is a really interesting group and probably important for understanding the origins and evolution of Bembex as a whole. And so you can see it here coming out as the most basal group. You also can see a little bit about some of the biogeography of Bembex species. It was thought that it originated probably in Southwest North America, coming out of there, and eventually goes on to become most diverse in Australia, even though none of its relatives are there, which is a very interesting history. But there's some further things going on here, is that this group, which is so important for understanding Bembex, probably has a lot of unrecognized diversity. And the females are nearly identical in this group as as in pretty much all other bembex. So it tends to be pretty difficult to distinguish those. The males are very easily distinguished from other bembex on the basis of those preserved ocelli, as well as other unusual structures like having a dentate labrum and some other things going on. But they're not easily distinguished from each other. And they tend to live nearly sympatrically. So there's a lot of different species that we've discovered that live in very similar areas, but technically not, because they all have different soil preferences. So they're living in different microhabitats and sort of doing their own thing. But when you sort of pull them up in a net and you just say, oh, these have the weird preserved ocelli, this has got to be this one species. They all get lumped together. There's probably quite a few unrecognized species that need to be described in this group. I have a couple if I ever get around to describing them. So this will be a really important group for understanding further evolution of bembex, especially this one particular species, Bembex magdalena, which is from the Magdalena Bay in Baja California. So it's really weird for bembex in that its terminal abdominal sternite of males has a sort of trident shape to it, which no other bembex has, as far as I know. They all have more of a tortilla chip shape. But this is a common state in the closely related genus Rubrica. So it's a really interesting question that you have this species coming out of a group which is probably the most basal in bembex and showing these characters which are really strange for the group but are found in other related groups. So this would be a really important species to study further. Unfortunately, it was only collected once, 12 specimens in 1926. Hasn't been seen ever again. The female is totally unknown, even though we can probably assess that it looks just like all the other females for the group. Still, it would be nice to have. So if anybody is ever collecting in this area and sees some wasps, please know. So one other species group that I wanted to discuss before I get off my soapbox for a while is Bembex cinerea and Bembex hyena. So these are really rare species restricted to narrow strips of saline coasts in North America. Morphologically, they're distinct from other North American species, and they're also distinct behaviorally as well. They do that sort of weird Bembex behavior where they do the truncated progressive provisioning, which is where they sort of do the typical Bembex thing of taking good care of the larva and putting in food every day, and then suddenly they jam all the rest of the food in and seal off the nest, which is probably an adaptation to the fact that their nesting sites tend to get flooded with salt water because they're right on the coast. So if that water is going to be coming in soon, you want to jam all the food in that you can, seal the nest, and just get the thing over with. But according to the genetic phylogeny, it's not closely related to the rest of the North American bembex. It's within otherwise Palearctic state, and it is coming up on the North American coasts. So I think this might be a really interesting species for understanding how the biogeographic distribution of bembex arose. It is interesting that the coastal species tend to be so unusual and genetically different, which does suggest that the coastal habitats could be important for dispersal and speciation. You see this a little bit on California's Channel Islands. The common species found throughout America, Bembex americana, exists on those islands, but it's started to diverge into multiple island-specific subspecies. So these islands are probably going to be a nice little laboratory for studying how Bembex, maybe how it got to Australia and started doing its weird stuff over there. The Australian Bembex are really weird. But speaking of islands, so I can go back to Honolulu for a little bit. That's my my great transition between different kinds of underground insects. So the second part of the talk I call Life in the Lava Tubes. So we are going to Hawaiian lava tubes, where for some time it was thought that there would be nothing to talk about during this talk because it wasn't expected that cave-dwelling arthropods would be occurring in Hawaii. There's a couple of reasons for that. One is that lava tubes are, geologically speaking, relatively young. So a lot of the mainland caves where you find these cave-adapted spiders and other arthropods have been around for much longer. So there's time for these things to evolve. But it was thought that the Hawaiian lava tubes were just around for too brief a time for that to happen. Additionally, the typical cave resources are not available in Hawaii. For example, bat guano, which provides a major resource or rushing water, which comes into caves and sort of drops off nutrients. These things aren't present in Hawaii. Hawaii has one species of bat, doesn't tend to occur in lava tubes. And lava tubes are much more fragile than a mainland limestone cave. So if water flows through them, sure, it brings in nutrients, but it will also collapse the lava tube well before anything has time to really take advantage of those nutrients. Additionally, the typical ancestors you would expect to do a very good job of moving into caves and diversifying there aren't present in Hawaii, which isn't much of a surprise because the sort of animal which would be pre-adapted to hunkering down in a limestone cave and doing its thing there is not necessarily well adapted to getting to Hawaii in the first place. But in 1971, in Hawaii Volcanoes National Park, Frank Howard, who I believe is in the audience virtually tonight, so please correct me if I get anything egregiously wrong, discovered a succeeded plant hopper and a cricket within these lava tubes. So there were, in fact, very obviously unique forms of insects in Hawaiian caves, and these are very obviously cave-adapted life. So the question is, what are these things doing in Hawaii where there was thought that they shouldn't be existing in the first place? Well, it turns out that while the typical mainland cave food resources are not available in Hawaii, there is a sort of Hawaiian solution to that, and that would be the ohia tree, which occurs sort of on the surface of these lava tubes, and its roots actually grow down into the lava tubes as they start to colonize some of the first things to come back after these lava flows. And these roots grow down into the caves, and it turns out that if you're a plant hopper and you get stuck in these caves, you can be feeding on these roots. And if you're a plant hopper feeding on those roots, well, something else getting into that cave will be very happy to feed on you or to scavenge you after you die. So over 50 years of surveys, and more than 60 lava tubes, it turns out there's quite a lot of cave-adapted life happening in Hawaii. So 45 different cave species have been found over what Bishop Museum's project, which I'll talk a little bit more about in a minute, calls the 11 core taxa. So there's plant hoppers, assassin bugs, springtails, some really interesting spiders. I'll show those. So there's quite a lot going on in these lava tubes where nothing was supposed to be happening at all. And this collection is represented at Bishop Museum as the Hawaiian Cave Arthropod Collection, or HCAC. So this is the largest collection, of course, of Hawaiian cave life. It's the founding collection. And it's a really valuable collection. Most of these species are rare or endangered because these environments are very fragile. There aren't a lot of individuals for any of these species at any given time because even with these roots growing down, that's enough for life to survive. It's not necessarily enough for a tremendous number of any of these things to be occurring at once. So this collection is very valuable and much is likely irreplaceable. So this is one of the really interesting species. This is a cave wolf spider. Wolf spiders are sometimes called big-eyed spiders, but this one has evolved to lose its eyes because it turns out they're not very helpful when you're in a lava tube and there's no light unless an enterprising scientist is down there with a headlamp. So these are commonly called the no-eyed big-eyed spider, which is a great little oxymoron. But in case you were wondering, there's also a little-eyed big-eyed spider, just for additional fun. And these tend to feed on things like the Hawaii cave amphipod, these nice little blind amphipods, which are scavenging and feeding on bacterial nets and things in these caves. So while I was at Bishop Museum, I joined a project which was initiated by my predecessor, Jim Boone, to recurate and establish this collection as a fully digitized resource for understanding Hawaiian cave life. Hawaiian cave life turns out to be very important because it sort of totally changed our understanding of how cave life could originate. It obviously occurred much faster in Hawaii than it did elsewhere. And it inspired some really interesting discoveries across the world. There were some researchers in Australia who said, hey, if they were finding plant hoppers in these Hawaiian lava tubes, there might be some in these similar environments in Australia. So they went looking, and sure enough, they found their own Australian plant hoppers. So this collection is focused on Hawaii, but it really has worldwide implications. So Part of what I did there was to start addressing some of these challenges. So there are some challenges for this collection. There's been a long pause in active curation. So obviously it's a mostly alcohol collection. These specimens tend to be rather fragile coming out of caves, so they need to be stored in fluid. When you're not tending to fluid specimens for a long period of time, that tends to evaporate. So that was something we needed to address. Lots of identification that needed to be done. Very exciting because a lot of these things are probably new to science, so a lot going on there. So this project was involved with identifying these species and inventorying them as well. So this project is currently going on through an IMLS Museums for America grant. So we're not just re-inventorying them, also digitizing and imaging them to sort of fully establish this as a research collection that's accessible for people throughout the world who are interested in working on Hawaiian cave insects and spiders and amphipods and things, as well as to establish a checklist of Hawaiian cave biota so that this is available for researchers. So at the time that I was working on this, we made some very nice progress. So in addition to the specimens themselves, we also have all of Frank's field notes centralized. So we're digitizing those as well. So the specimens are currently all imaged. We're working on getting those digitized, all the field notes available. So at the end of this project, what we'll hopefully have is not just a full list of Hawaiian cave life in all of these specimens, but also the original notes that went into these collections and understanding where these things are coming from very specifically. So there are some significant challenges for this as well. So these cave environments are very sensitive, not just ecologically, so they're very fragile, they tend to collapse. They're very localized, so these populations may be extremely small. They sometimes can move around between lava tubes through cracks and crevices, which are too small for humans to certainly to get through, but also to see if they're even there, but a Hawaiian cave spider can do it. But they're also sensitive politically. Many of these are on federally protected land, others are on private land, and both probably don't want people tramping about willy-nilly to try to find rare Hawaiian cave insects. So this has some pretty significant consequences for data generation and management. One of the things that is usually great to have on a digitization project like this is to do crowdsourcing. So you have all of these collection labels, and when your volunteers are starting to get burned out, typing up all of these lava tube notes, then you can put them online for people to take a look at. But if these are locations that need to be kept in confidence, you can't do that. And also, we're going to put all of this online, which is Bishop Mizuno's goal, but there also has to be some data scrubbing to ensure you're not publishing the location of federally protected species. Those species of cave spiders are on the federally endangered list. Most species that are coming out of there will probably need to be taken care of in some way. So there's pretty significant challenges there. But there's also some really great opportunities. It demonstrates the importance of these cave environments ecologically, not just for tourism, not just for the study of the geology of these caves themselves, but also for this life that was not supposed to be there. There's also really fantastic outreach potential for the Native Hawaiian education programs, for the public to really get people not just to understand that these things are there, but really to appreciate them as a sort of unique facet to Hawaiian life. I was only in Hawaii for 2 years, but one thing which is readily apparent to anyone who spends time in Hawaii is that there's a lot of passion and a lot of pride for the native plants and animals of Hawaii. But these are some which are really unique and really not well known, but they are tied to the Ohia trees very intimately, and those are extremely important as symbols in Hawaii, as well as ecologically. So there's a lot of opportunities there. And I think if I have one take-home message from this talk, which is sort of generally grouped by Insects Underground, but it's also sort of cool things I've done in the last 10 years or so, it's that there's a lot of interesting things that are happening just beneath the surface that have consequences, not just for the very small microhabitats where they're occurring, but across the world. For instance, sand wasps that you're studying out in, say, a baseball diamond in Illinois, where for some reason there's one species that really likes the sort of texture of packed soil in baseball diamonds, might have relevance for understanding why wasps in Australia are doing these sort of unusual things like attacking damselflies, or studying the cave insects of Hawaii can have significant consequences for understanding cave insects across the world. So please pay attention to things that are lying just under the surface, both literally and metaphorically. So with that, thank you very much.
I have a question from the chat.
All right.
How do you associate males and female fins?
So the question is how male and female bembex are associated. So that would generally be by collecting them together. So you would have to collect them within the same event. That's the easiest way to do it. The males are readily distinguishable. The females collected in association. But while the females tend to be very compact and it's a little bit awkward to identify them based on color, it's not impossible. So you can identify the females if collected alone. It just tends to be harder, and they're certainly not showing as much variations.
What was your success rate for extracting DNA and successfully getting UCDs from your legacy specimens?
So
I forgot the other half of my question.
That's okay. I'll go off with my answer and maybe you'll remember. So the question was what my success rate was for taking usable ultra-preserved elements out of these archival museum specimens. The success rate was about 30%, about 1/3, which was pretty good. Some of these specimens, the average age was 40 years. I went back as far as 90 years and as recently as about 20. So about 1/3 was pretty good, I thought, for those specimens. And this was extraction from 1 or 2 legs. It probably would have been more successful if I was willing to grind up whole organisms, but I do have an emotional connection to these specimens, so it didn't really work. Yes.
So you talked about behavioral plasticity in the way that they're excavating their nests. I was kind of wondering, is there any data available or any general conclusions that can be reached on how they're adapting their nest digging behavior to like the soil that they're in? Like you talked about different populations do different things, like some of them have spurs, others don't. Does that have anything to do with the kind of soil that they're in?
So the question was whether the plasticity in bembex nest behavior is related to the soil and microhabitats. To some extent, it looks like it may be, but the thing is that the real driver of bembex nest diversity appears to be parasitization pressure. So they're dealing with velvet ants and satellite flies and things that try to get into the nest and either eat the provisions, eat the larvae, or both. And a lot of these structures seem to be related to trying to avoid predation or parasitization. For instance, these back burrows, they seem to serve as sort of a false entrance to the nest, so that a mutilid, which is trying to get in, will waste its time digging into something that has no bembex larvae beneath it. Other structures seem to have no clear purpose. For instance, the nest spur might be used for the females to sleep in, but they clearly can sleep elsewhere because not all species have a nest spur. I believe there's some evidence that the depth of the nest may have some connection to the temperature of the soil. And as I mentioned, for the coastal species, that truncated progressive provisioning behavior is probably an adaptation to the fact that the nest is going to be overrun with salt water sooner rather than later. But mostly it appears like it's an attempt to avoid the nest contents being eaten or on in the future.
Pretty interesting. Thanks.
All right. Yes. With regards to sympatry, you mentioned that some species have been found in the same region or like small microhabitat, but in different soil substances. So are there species that are found in the same soil substance?
So the question is whether there are species that are found in the same soil habitat. Yes, there are. The differences between these soils can be really pretty small in terms of texture. In some cases, they're being found in the same sand dune, but they like different parts of the dune. So in terms of geographically, they really are being pulled out of the same area, but the soil differences are going to be very, very slight but present. But others have relatively unrestricted soil preferences. So they might be okay using nesting habitats that other bembex would. But the populations tend not to be truly mixed in that you'll find a nest of one intermingled directly with the others. That's not something that's usually seen.
And have you like compared the specificity of that with your phylogeny?
I have not compared that directly. And part of the reason for that is that the species that I was able to get DNA from don't necessarily correlate with the species where there's really good soil data. Some of the observations, especially made by Howard Evans, are really fantastic in terms of soil temperature and consistency, but other species, we either don't know the behavior or there's very general observations of the soil. So I think really taking a look at the microhabitats would require a very close examination of soil at time of collection, which if anyone out there knows a funder who wants to throw money at someone to do in their spare time, please.
In regards to the Hawaiian cave arthropods, were those on different islands, or what is the pattern of diversity if they're found on more than one?
So there's sort of two patterns of diversity. So these caves are found on different islands, but the diversity pattern tends to be regulated much more by the proximity of the lava tubes themselves, because it's not easy for there to be dispersal between them. So there would tend to be a lot of differences. Certainly they're not, once they're getting into these caves, moving between islands. So mostly it's a very small scale pattern of biogeographic events.
I'm curious technically how one studies the architecture of nests built in sand.
So the question is how you study the architecture of nests that are built in sand. And you actually do a technique where you dig not the nest itself, but you dig next to the nest. And then you try to break into the nest from the side. So you're preserving the structure of it because you're not just scooping the whole thing out, but you're very delicately following it down. And it's not easy. I've tried it myself. But you can sort of imagine it's a little bit like chiseling away at a rock, if the rock was extremely soft, and trying to get at something in the middle rather than digging it out directly. And you can sort of pour a white or brightly colored powder down the nest entrance to help you track where that is as you're going inside. But it is sort of a side excavation. But yes, this is made of sand, so it will collapse very easily. Bembex wasps like to dig their nests where the soil will be enough to keep them intact during their normal digging operations, but they're obviously digging at a much smaller and much more gentle level than a big clumsy human with trowel would be doing. So it is pretty tricky, but it can be done. It's not something where you have to pour liquid metal down it like you would for a big ant.
That's a you.
Thanks. While I was thinking about that question, I heard is a possible use of the spurs in the nest as a place to catch any falling sand that may be disturbed down the canal, but like rather than filling up the nest, it will be caught in the spur. So,
yeah, the question there is whether the spur is used as sort of an accessory soil storage, and that has been proposed. I don't think it's been shown one way or the other. The general theory is that it's sort of used as either a sleeping structure or it just sort of develops incidentally as the wasp sort of pulls soil out of another part of the nest. But it is possible that it's being used as a clearance. There's I mentioned earlier, there's closures to the nest, multiple closures. And the inner closure is just at the transition point between the nest cell, which is where the larvae and food are kept, and the spur. So it may be that in species that make the inner closure, that soil is being stored there. It's not for a nest collapse, but for a deliberately built structure. But the correlation between its inner closures that has a spur is not 100%. So at the very least, it's not always being used.
So I had a question. How did these nests survive being flooded by salt water?
So the question is how these nests survive being flooded by salt water. So there's sort of two ways that's probably going on. One is the structure of the nest itself, which the water would sort of hopefully compact the soil. So it's hopefully keeping that tubular structure intact. And I believe that those nests aren't terribly deep, so there's not a tremendously long structure that may collapse. But there's also probably some resistance of the larva would have to be to, at very least, salinity in the soil, even if water is not directly flooding it. So I don't think this research has been done because these species are not terribly commonly collected. But it would be very interesting to see at what stage of larval development they're surviving if the water is flooding. If they're in their cocoons, they might be relatively well protected from water flooding them. If they're still a soft exposed larva, they might be in bad shape, or they may have some sort of resistance to salinity. So that work hasn't really been done. These are very interesting species. So hopefully that work will eventually get a chance to shine.
Jeremy, I've got a question about the coastal species having weevils that are on the coast. What is their conservation of outlook?
So the question is, what is the conservation outlook of these coastal species? I don't believe anyone has really checked on that. I haven't seen many specimens of those collected, certainly not recently. They're restricted to really narrow strips of coast from Florida up through New York. And so they're only occurring at the tide line on beaches. As you can imagine, that these are popular spots for other animals like humans, even though there aren't really any other insects there. So I'm guessing that the conservation outlook probably isn't good. They've been rare since the 1950s when these observations were first being made, and haven't seen anything on that since. So hopefully they're still around. But
Well, you're on the East Coast now, so maybe you can go check.
Go pay a visit.
Yeah.
This was kind of following
Hi,
up.
Jeremy. Hi, Jeremy. Frank Howarth here. Thanks. Very nice overview of the Hawaiian cave fauna. I would add that my studies in Hawaii showed that the cave animals could live primarily in the smaller cracks and only occur in caves when the physical environment is suitable. So populations are probably much larger than what we assume. But you're right. The species that we've studied intensively on the Big Island, they do segregate out into geographic but distinct populations. We have 9 or so species of Olearis polyphemus sensislatus, the picture that you showed, and by the vocalizations, the sounds, the mating songs they produce, and slight morphological differences, we can separate them out. And DNA studies confirms that we can separate them out into distinct populations. I would also add that the discovery of the cave fauna in Hawaii and the fact that they're close surface relatives completely upset the standard paradigm of the evolution of cave animals, and now the confirmation that the tropics hold the vast majority of cave species, which were mostly unknown before the discoveries in Hawaii. So now there's a big effort to survey tropical caves, and the number of species is going up exponentially. And in addition, the studies of the crack and crevice fauna, where it can be accessed, it shows that it's that's equally diverse. So so we've now these discoveries have improved have it demonstrated that the caves are ideal laboratories for evolution and ecological research, not only for cave species but for evolution in general. So we've refined the science of speleology. So, but very good job with an overview. Thanks.
Oh, thank you. I really appreciate your insight.
As a follow-up to that, Don Davis in Zimmerman, 1978, has also reared a related species, Ferroica alutella, from larvae collected on pigeon dung in Pigeon Cave, Hawaii. Pigeon Cave.
Killing the role of rats.
For some reason, that's included in our publication of new species and records of moths from African caves. I don't know why it's in there.
Pigeons tend to pop up better, including caves and seemingly in early as well. All right, any other questions? All right, well, thank you very much.
Thank you, Jeremy, for your excellent talk. And if no one has any objection, I'm gonna adjourn the meeting. Would you like to move that we adjourn the meeting?
I so moved.
Second.
Oh, there's CC as a second.
Can we discuss it?
Not discussable. All in favor, say aye.
Bye.
Any objections? So ordered.